1 minute. All right. You're moving. Oh, I'm ready. All right. Welcome back, everyone. Thanks for coming on back. I think I hear some tummies grumbling for lunch afterwards. All right. So my name is Dustin Robinstein. I'm here at the Biotech Center at the Advanced Genome Editing Laboratory. And I was asked to talk a little bit about how we use the digital PCR instrument, the Absolute Q. So I'm going to my plan is to kind of give you a rundown of, some use cases. So how do we use it to know exactly, to better understand the models that we create. But before I do that, I did want to do a quick introduction to biotech's new BioGarage. And I think it's actually a pretty exciting. What is the BioGarage? I think it's about three different things. Number one, some really exciting walk-up instrumentation that's available. This is a model that, I think you're familiar with at Course, right? I mean, instrumentation you need, schedule it, pay for it, schedule it, use it, and pay for it. Another category involves some services. So maybe some things that you just really don't want to do and you can sort of offload to someone else. But I think one of the really exciting things that BioGarage can be is a fully equipped molecular biology lab space. So there are a lot of labs on campus that have ideas and have maybe are equipped for protein or maybe don't even really have a significant lab space. But the idea would be that for a certain amount, you can rent out open bench space in the BioGarage and have access to a fully equipped molecular biology lab. So if we just first look at instrumentation, we've got instrumentation for RNA and DNA preps. We've got a QIAcube Connect that does this automated RNA DNA prep processing. There are other tools to do more traditional processing if needed. I believe we'll eventually have a gel electrophoresis and imaging setup there as well. To QC your RNA and DNA, we've got a bioanalyzer and a QIAxcel Advanced. And like I said, eventually, hopefully gel electrophoresis, I think. And then, of course, to analyze your samples, we have the Absolute Q here, like we've been talking about. And to Holly's point, digital PCR isn't always the right answer. Sometimes qPCR is still best, especially for those sort of workhorse, RNA expression studies, for example. In that case, we have 3 Quant Studio 7 Pros available to you. So these are instruments that are available. There is a tool online somehow. If you go to the BioGarage's website, it'll allow you to book these out and use them as you see fit. Yeah, actually, I'll get to those other ones in a second. Workhorse services is still kind of something that's sort of developing. I think we're trying to figure out exactly what are the services that would be of use. I think a couple obvious ones. Number one is a next day mouse genotyping assay, right? So you have some pups, you want to sample them, you want to know very quickly exactly what genotypes you're looking at. We're putting together a program in place so that we can get something like either same day or something like next day, essentially mouse genotyping available to you. Additionally, plasmid preps of different kinds. Some labs are or are not equipped to do that, and that's something that would be available. But the idea would be to, you're an expert in something that's probably not mini preps, right? So spend the time doing the things that you're an expert in. But to me, at least, I think the most exciting thing the BioGarage could be, and maybe many of you, this is not appealing because you already have a molecular biology lab. So maybe this is not exactly the right audience, but I think for many folks, especially those practicing, clinician scientists that don't really have a set up molecular biology lab space. The idea is that we have open bench space. We have all the tools you'll need to perform your experiments. And for I think the thought now is an introductory rate of about $1,000 annually. You have access to is all the absolute Q you can eat, so to speak. And the idea would be that so all the instrumentation that I mentioned, as well as room for growing bacteria and harvesting plasmids and stuff and cloning, as well as a tissue culture space, which is exciting. So BSL2 got all the things you need: biosafety cabinet, incubator, all those things. So if that is something of interest to you, don't need a duplicate of that. Feel free to send an email to biogarage@biotech.wis.edu or go to the biotech website and you'll see one of the obvious, one of the cores is on the BioGarage. So feel free to reach out to these folks and if that's of interest, as well as to use the absolute Q, I should say. So for digital PCR instrument usage, down the road, this would be your contact. Okay, so now to kind of get into, why I'm standing here in the first place, and that is to give you a sense of how we use digital PCR to better understand the models we make. So I'm going to start with, I guess, just telling you just a real quick bit about what we do here at Agile Advanced Genome Editing Lab. We create genome edited cell lines. We create genome edited mice, rats, and pigs in collaboration with the Animal Models Core here. We help labs do pooled lenti CRISPR screening if that's of interest, as well as other perhaps exciting, advanced custom CRISPR applications. That might be in vivo editing, it might be some other clever ideas that have come up. So, I'd say overwhelmingly we're creating these models, but we spend a lot of time and I think the biggest value added we have is probably on some of these other things as well. So when it comes to creating models, anyone who spent time doing CRISPR, I'm not telling you anything you don't know, but just kind of setting the stage with the things that we'd want to do. Some of the key challenges when you're creating models is, number one, on target confirmation, right? So I made this edit on target. Is it exactly what I think it is, or am I wrong? Number two, sometimes you just want to know, estimate your editing rate, either, some indel generation, or you want to investigate your knock-in rate. And then, of course, off-target analysis, right? So those of you, how many, just real quick, show of hands, how many are doing genome editing now? Or, yeah, okay, right. Okay, it's pretty, so many of you are probably aware of these sorts of things and the needs to do those. So I want to give you, I'm going to go way back to 2017, 2018, something like that. Brent, I don't know. It's been a little while, but this is kind of a good use case for when we sort of first figured out that the gold standard in the field generally is PCR your target and sequence it, right? But if I cut to the chase here, sometimes that's not enough. So I'm going to give you a little story of exactly kind of how we came to that point. And this is even before compartment-based digital PCR instruments were available. So we were using the BioRad at the time. So we had already created lines of pigs that carried mutations in this NF1 gene, which is we're studying this neurofibromatosis type 1, a monogenetic disorder. It's fairly complicated. It's a wide spectrum of disease manifestations. And we're creating pigs to best model that disease. And we were looking to take a couple founder pigs that we generated that carried a particular mutation that we were interested in. Now, for anyone who's worked with mice or rats, the first step you do is you take your founders and you backcross it for a generation. That way, they got exactly one normal allele from the unedited parent and then the edited allele from the founder parent, and you can kind of isolate your chromosome that way. You're good to go. Of course, that's a luxury that isn't really available so much in pigs because, they're giant and they cost a lot of money and it takes a lot of time for them to become giant. So we're really looking for a way to skip that outcrossing step that necessarily gives you hets. So we're looking to just breed founders. But what we saw, for example, here's a case where we had one founder that was, let's say it looked, in the case of at the expense of oversimplifying, let's just say it was a wild type pig that carried mutation NF1A, and we bred it to another pig that was wild type and carried NF1 mutation B. Now, of course, what we'd expect, what we're hoping for is to get A's over B's, right? But instead, what we got were these crazy pigs here. This is not what we were expecting. We did the PCR and sequencing, and these pigs looked like they only had this one sequence, just NF1A. Now, of course, that shouldn't happen. We didn't see allele A in this pig. So that got us, first of all, question the very fundamentals of genetics and how we came to be into this field. And once we kind of took a deep breath and calmed down, we realized that maybe what we were measuring is not what we thought we were measuring, right? And to give you a sense of what we were measuring, let's take a look here at the assay we did. So this, when I say NF1A and NF1B, what it was is a deletion of this alternatively included or excluded exon A31. So what we did was a couple of digital PCR assays. One of them just interrogated this cut site here, right? So forward primer with this R1 primer and a probe set right here. And this allowed us to count every single allele that was not excised. Then we created a separate assay, which was for the excised allele, right? So now we were going from essentially from forward here all the way to R2 over here with that same probe. So this allowed us to count all the excised alleles, right? So you would imagine that either, all the alleles, an animal is going to have two alleles, and it should be some combination of not excised or excised. And when we just do the simple PCR, we saw some that carry a couple different bands, so they had excised and non-excised. And here's an example of 4 that all we saw was this is just conventional PCR, right? You just run a gel, PCR and run a gel. And we saw that these 4 samples here only gave us the excision size band. So what we would consider as homozygous for the excision. So now what happens when we take this setup that we used to quantify each one of these alleles separately? If we look at the unedited, so the gray is this here and the red is this one here. The unedited, we see essentially two copies here. For we have an actual heterozygous excision where we see one allele here and one allele here. Great. A couple homozygous excisions, right, where this is 1.8, 1.7, it's about 2. This is with droplet-based PCR before compartment-based digital PCR existed. So those look to be truly homozygous, but then you get these samples down here where we see one allele of excision and nothing else, which led us to the conclusion that there's some other allele that we're not capturing with these assays, some unexpected genome chromosomal rearrangement event that is just not what we're expecting. So these animals are not pure heterozygotes. There's some sort of compound het carrying an excision that we'd expect and some sort of cryptic mutation, what we call cryptic mutation, something that just isn't readily identifiable. And now we live with the fear of cryptic mutations all the time. So in that way, it allowed us to have a positive identification of these cryptic alleles, right? So rather than it just being this thing, this spooky thing that you just can't identify via PCR, now we have a method to know for sure that we're missing something. We don't know what it is, but we know we're missing it. Okay, another use case. This is fairly early on when we were looking to create A set of mouse models for a user. There's this region in the genome that has a couple orthologs, these gene duplicates here. And they're interested in redundancy, so they wanted gene A knockout, gene B knockout, and the double gene A, gene B knockout. And of course, the easiest thing to do would just be to give these mice, both targets, gene A, gene B, find some that have gene A and gene B knocked out, find some that have gene A and not gene B, and then find some that have gene B and not gene A, and bingo, right? Easy peasy. So that's what we did. We gave all the mice both targets, and then back-crossed them to a mouse that hadn't been edited, right, to extract those alleles. But these were maybe around the same time. I'm not sure. I think it was around the same time. Thanks, Brent. My old memory is escaping me sometimes. So thanks for the help, Brent. So we actually thought about this for a second. So we back-crossed them, and the idea was, okay, sequence gene A, sequence gene B. The simplest approach would be find the mice that have just mutations in gene A, bingo, those are the gene A. Find the ones that have a mutation in gene A and no mutation in gene B, bingo, those are just the gene A's. Find the ones that have no mutation in gene A, but a mutation in gene B, bingo, those are the alleles that just have a mutation in gene B. But then we thought about that for a second, and maybe that's not exactly the right assay, because you can imagine that maybe there's two different alleles here, right? Let's say we're trying to get the single gene A knockout. We PCR and sequence this. In this case, we're going to PCR and sequence, and we'll see that they're het for the knockout. This is what we know and what we'd expect. And in this case, if we were to PCR and sequence, we'd get all the sequences back, and they would be just the reference sequence, right? So that's great. But what about this other scenario, again, where we have a knockout in gene A, nothing in gene, and the reference, other allele is a reference gene A. We'd PCR and sequence, we'd see it's a het, great. But what if the other target site was actually some invisible cryptic mutation again here, where we PCR and sequence, and we think we're amplifying both alleles, but because there's usually what we know now, it's almost always some massive deletion, although I guess sometimes it's a massive insertion even from other organelles. Okay, so I won't limit the creative double-strand break repair process to just say it's excisions. Any weird thing can happen sometimes that blocks PCR. So the other possibility here is that we think we're amplifying both alleles, but really we're only amplifying this allele, and this one is invisible to the process. In using just PCR and sequencing, these two are absolutely indistinguishable, right? So, oh, I don't have the data here. Okay, well, we used a similar design, to this, essentially, apologies, to test this and identify the mice that are pure gene A in unmodified gene B versus some that are, cryptically double mutation mice. And we're able to cleanly separate those and hand those off to the investigator, which have been working out well. So after these couple of use cases, we were realizing that digital PCR is probably something we're going to have to incorporate more often. In the Bio-Rad droplet digital PCR worked, but it had some limitations. Number one, it's droplet based, right? So you have to go through this synthetic process, this emulsion process to set up your PCR, set up the emulsion, then run the PCR, then run the emulsion after that. And it was quite tedious and labor intensive. And I think, Brent could always know when that was going to happen, because if it was like a horrible blizzarding day, he'd say, let me guess, this is a digital PCR day, because there would be like 6 inches of snow, or 6 inches, 2 foot and 6 inches of snow. Anyways, we seem to pick the worst weather days. But that just emphasizes that it's a lot of, shuffling around back and forth. But even more technically, you have to be able to reliably create these emulsions of your PCR mix. And if you have things that are unexpected, when I say unexpected to the master mix or unexpected to the chemists that design this, it might throw off the ability to emulsify these, right? If it's something that could break some sort of detergent or whatever the opposite of a detergent would be, disrupts the ability to create this emulsion, and it really disrupts your ability to have any kind of precision with digital PCR. And since a lot of the times at this time, we're starting to do more and more kind of quick and dirty PCR, for example, I mean, who really all the time wants to have like hyper pure DNA and measure that, right? So sometimes for cell lines, we're just taking a little bit of media, bringing it up to 95 degrees, killing the cells and using that. And like, so, but there can be weird stuff in your mix, and having to always worry about like whether that's affecting the emulsification was not ideal. So we really wanted to focus in on one of the partition-based platforms. And at the time, there was the Absolute Q and the Qd. We were thinking about doing that. We started doing this in July 2020, and some of you might recall that there was like this like global pandemic going on at the time. I'm seeing some nods. I think some people remember. So having someone, just like throwing your doors open and having visitors come in and just like, plop down an instrument so that we could demo it, that was not an option. We were all, as we were all hunkered down, maybe in the lab, if not at home. So what we did is we took the same panel of samples and assays, and we sent one to QIAGEN, we sent one to Thermo Fisher. Well, at the time it was Combinati, but it's Thermo Fisher now, same instrument. And what we found, we sent them essentially two assays. One was kind of just like a workhorse assay, just like how does this, just something that worked fairly reliably and just want to know how does it handle that. And then another one was a sample of, we call it mixed purity, or maybe it was not as pure. So I think this case was one of those just cells in media, and we just, lysed it in media and asked how well do they each perform. So the Absolute Q actually gave us meaningful data with that crude sample prep, whereas the QD, there's no data output that made any sense really. And even with the standard assays, we were finding just more robust, cleaner outputs from the Absolute Q. So at that point, we decided this is just less of a headache for us, especially because as a core, we're often going from project to project to project. So it's possible that with optimization, maybe they would have worked equivalently, or maybe we just were anomalous for other reasons. So I'm not telling anyone you should do whatever works best for your group, right? But for us, optimization is like a curse word, because we're generally going from like project to project to project. We might run it a few times, then move on to the next. So we needed things that sort of worked reliably with no optimization, and that was sort of essential to us. So that seemed to be true for the Absolute Q. And finally, Holly mentioned this, that the plate usage is really flexible. We don't usually have 16 samples at once. We usually have 4, maybe 8. So you can use, a single column at a time and then keep the plate and use the rest. So that being said, we went with the Absolute Q, and that's what we have here at the BioGarage now, available for you. Oh, sorry, ignore that. That title is not right. So what I wanted to talk about here was confirming homozygosity in cell lines, for example, right? It's quite easy in animals or some other form where you have, sexual reproduction and independent segregation, but in cell lines, you just have what you have, right? And we talked about how there might or might not be a mutation in this gene that you can, in this region that you can detect, or maybe you cannot detect. So to just PCR and sequence it is not sufficient. You really, if it's important for your experiment to know that they're homozygous, then you really need to do more than just PCR in. So what we do is we do PCR and Illumina amplicon sequencing, and that's really just not enough. So what you can do, for example, in this case, we looked at unedited cell line and then a couple of clones. Those clone names might look familiar to some in the audience. We were able to confirm that these are, in fact, homozygous knock-ins in this particular case. Another application here. So I want to talk for those of you who have done any sort of large knock-in or any kind of knock-in whatsoever with CRISPR, are aware that if you're going to knock something in, the overall screening strategy is that you put one primer within the knocking construct and the other outside this homology arm, right? So the primer that's outside the homology arm gives you site-specific information, right? So you know that your construct actually went in at the targeted locus and not just off-target integration, which happens often. So in this case here, what we had is a particular target, and we had a knock-in construct. The homology arms are highlighted in blue here. And what we expected was that the single strand, that you get single strand annealing here, you would get extension across this knock-in cassette and into the other homology arm, at which point it would come out. That's the way we all draw it up in our head and in the cartoons that help us go to sleep at night. But it turns out that we had recently had a project like this. Maybe you've seen in other cases too, but where there were some things that were just kind of starting to look a little funny, that just seemed to be a little off. For example, the homology arm PCRs were a little weaker and not as robust as we'd like. We were seeing some unexpected C and V values using digital PCR that kind of led us to think, well, maybe there's a bigger problem with what we had. So we did long read whole genome sequencing with Oxford Nanopore here at the Biotech Center. And what we found actually was that when we were PCRing this and PCRing outside the homology arm and then PCRing, from reverse from within this knocking cassette, and then doing the same thing here to here, this wasn't the case. Actually, what happened was we got single stranding and annealing, again, as you'd expect, and the extension went across the knocking cassette. But instead of getting on the off ramp here, what it did is it just kept continuing going around the plasmid, not once, but at least twice before coming back out. And because of the read lengths we had, we don't really know exactly how many times it went around, but it was at least twice. So what we saw was something more like this. But digital PCR was something that helped us to identify that, without getting too deep into it, that there was something kind of funny here and that we probably need to look a little more closely. Another thing I wanted to tell you about, so I talked so far about how to know that your alleles, that the on-target mutation is what you think it is. But sometimes, as those who have done genome editing, you really just want to know, what's the ballpark efficiency of this edit? And sometimes that's really easy to do with a number of methods, but and sometimes I'm giving you a case here where I think digital PCR was sort of the best case here. So here we look to create a large knock-in to create a nuclear donor cell line. So the idea would be to create to edit the Wisconsin miniature swine line to create a pig that has an inducible Cas9. So the design here was that to use GFP as a positive selection marker. So we use flow cytometry to screen for eGFP. But we started to get concerned that because this construct was so massive, what we really might be selecting for is just knock-in that occurred on this side, rather than across the entirety of the cassette. So we coupled the eGFP selection with the M-scarlet expression. And sorry, not expression, the GFP expression in flow cytometry with the M-scarlet C and V analysis. And what I wanted to show you here was that even though we had fairly low sample inputs, and this was a pretty quick and easy experiment, we still got really useful data. So these are across essentially technical replicates from a sample that was, this is not. My point here is not that this is like beautiful data. I'm showing you data that is kind of more like the data you get, right? This was with no optimization, just taking the sample we have, we threw it on there because we thought it might be a little messy. Brent luckily thought ahead and ran in a couple technical replicates, and we saw these pretty reproducible values that gave us efficiencies of about what we'd expect and were useful for us going forward. So I think what this would, if there's something I'd want to mention here, is just how really straightforward this is. This does not involve some long drawn out procedure. It's Well, it's easy for me to say because I'm not the one who sets it up, but Brent always tells me. It's pretty much setting up a PCR and throwing it in the machine, and then the data gets uploaded to the cloud, and you can sit on your butt from your lab and download the data. Okay, so I guess that's all I have for this. I think Lenny, were we gonna next do the talk about the okay, so what if sure. So why don't we do this? Why don't we, if first there's any questions now, let me give you a quick highlight. So I'll answer a couple questions now. Then we're going to have the Thermo Fisher folks come up and explain how this demo process will work. And then if we have time, maybe we'll do like a question and answer session, maybe help you think about applications or help you think through some stuff. And then once lunch comes, then we can do that. Does that sound about right?
That's good.
Okay, cool. So that being said, any questions on anything I just talked about?
Dustin, have you done this with your CRISPR experiments just to instead of doing knock-ins, but just looking at single point mutations at all?
Yeah, definitely. So so that's kind of exactly. So that's kind of what we're looking to do here. You're right. So sometimes yeah. So large knock-ins can be tricky because it just involves a large piece. Small knock-ins can be easy. You can usually PCR, but still, the point is, I think what you're getting at is sometimes you just never know, even if it's just a small, like what could possibly go wrong? It's just like a single nucleotide variant. It seems like as soon as you generate that double-stranded break, things can go sideways. So even for cases where you're just looking to create an SNV, I think if it's something like a cell line, something where or maybe an animal where you don't really want to wait tons of generations, then I think it's probably essential to do something like this. Or maybe for I think one of the other nice things about CRISPR is that it's so easy. It's sort of an afternoon like throwaway control. Maybe you don't really care. Maybe it's not. Maybe if you're right or wrong about whether it's a true het or a compound het, and you just have to weigh, like, if I'm wrong and it's not the sequence, how does it affect the interpretability of my results? It's not like everything we always have to do we have to spend all of the money in the world looking at it. You just need to be aware that these are the kinds of things that can happen, and then ask yourself, is it worth going to that next step to validate it? Any other questions? Cool. Thanks.
Great.
Let me, you guys want to share?
Sure.
You guys can share.
Thank you.
Hi, everybody. My name is Lenny Bain. I'm the genetic analysis rep here in Wisconsin. So that's okay. So May 31st, June 1st, we're through the help of the BioGarage, Dustin, who is apparently running the BioGarage now. Somebody is. Has opened up their doors to us to allow folks here or the folks at home watching, viewing, to contact us, set up a time to actually run samples, basically free of charge. We're providing the MAP 16 plates, the master mix. All you have to do is provide the primer probes and your sample, and we've got a protocol already developed for that. So just reach out to us, reach out to me. We'll schedule a time to talk to Holly, our field application specialist, make sure everybody's all on the same page. Because we don't want any surprises. We don't want everything to go smoothly. So, don't be shy. We're happy to do this for everybody, and it's helps everybody all the way around. Holly, did you want to say anything?
Do you have a copy of the protocol with you?
I do. I have a copy here. You can also reach out to me and I can email it to you, which the copy actually has some hot links on it. So it's probably a better way to go if I email it to you. That's about all I've got. Do you have anything that you want to add, Holly?
So after you've had a chance to kind of visualize some of the kind of results you can get from digital PCR, please feel free to think about how that would apply to your project. We did talk about transitioning easily from qPCR over to digital PCR. There's a variety of ways to do that. In this particular demo, we would prefer to work with you to give you the most clarity of information. So if you have an assay designed that is a probe-based, labeled with FAM or FIC, it's an easy place to start. We can transition that. If you have a project where you're working with cyber green, we'd like to walk through it with you and make sure to double check the parameters. We might make a suggestion to try a probe just for this particular run to see what kind of data you can look for detection, because probe-based technology is a one-to-one relationship, and so you get better clarity. We'll walk through all those details with you, make sure you have everything on hand before the demo, and then when the day that you arrive, it will be very straightforward just to have the protocol prep with your samples and load them into the go.
Did you want to add anything, Dustin?
Great.
Okay, sounds good. Thank you, everybody, for coming. We do have lunch over in the other room, so feel free to join us. Thank you. Oh.
Is there any questions or anything that they would like to address between the three of us?
We do. I've got some copies over at the table. Thank you. Thank you.
Thank you.